Hello, everyone, and welcome to today's The Scientist Book Club discussion. My name is Bob Grant. I'm editor-in-chief of The Scientist, and I will, as usual, be moderating our chat. Before we sink our teeth into today's discussion of Venomous, I want to remind everyone about the Scientist Social Club. This is a group on our Facebook page that we started last summer in order to find new ways of engaging with our audience, both scientists and the science-curious public alike. The club hosts conversations on our Facebook group, book club discussions like these, and other activities, depending on what we think up. We also hope for the social club to be very much a collaborative effort. So please make suggestions within that platform, engage in the discussions that we try to encourage there. And let us know what you'd like to see us do with the group of people that we have assembled and the platforms that we have available to us. So today we'll be discussing Venomous, how Earth's deadliest creatures mastered biochemistry, with author and molecular biologist Christie Wilcox, who is now the Scientist, I'm very happy to report, newsletter editor. We're so glad that you all have joined us for this discussion, which is sure to be engaging and eye-opening. Again, these discussions work best when the audience gets involved, so please do feel free to share your thoughts or questions directly for Christie. I can ask those of her as we sort of sift through them on the back end. At any point during the event, you can submit your questions or thoughts by clicking on the chat tab to share how you interpreted different aspects of the book, or head over to the Q&A tab to pose a question directly to Christie to answer during our session. Please also remember to let us know where you're joining from. It's always fun for me to see, this person is asking a question from Malaysia, or this person is asking a question from Indiana. And we'll try to give your locale a shout out as well. So after this event, the video for this webinar will be archived on the Scientist website. And we will send registrants a link via email within a couple days. So with that, let me introduce our speaker, Christie Wilcox. Christie was already a well-established science blogger and writer when she was awarded a PhD from the University of Hawaii in 2014 for her research on the genetics of lionfishes. Just 2 years later, she published her book, Venomous, How Earth's Deadliest Creatures Mastered Biochemistry, which received widespread acclaim. And after that, she fully left academia behind and established herself as a very capable science writer and editor. She joined The Scientist just this year in 2021, as I mentioned, as our newsletter editor, and we're thrilled that she's a part of our team now. I believe Christie has a brief presentation giving us a close-up look at some of the most lethal venomous animals in the world, so I'll toss it over to her now to show us some cool images.
Hi, everyone. Yeah, I never like talking about my book without the ability to show images, because I think venomous animals are some of the most beautiful animals, in addition to being deadly and amazing. In short, they're just incredible creatures. And they are so diverse, which is the other thing that just amazes me about venomous animals. I mean, you have venoms evolving in so many different clades, so many different groups, all different types. This is a photo I took, for example, of two scorpions who are very, very interested in each other. And yeah, I loved that for this book, I got to travel around the world and do all sorts of things to look at all of these incredible creatures. So one of the questions that often comes up when I'm talking about my book is just sort of, how did I get into venomous animals? And the short answer is that I've just loved animals my entire life. One of my favorite stories to tell, which is entirely true, and I have the receipts to prove it, is that when I was 5 years old, I told a teacher that I like to open the mouths of dead geckos to look at their tongues. So one could say I've been a biologist since I was at least 5. But more specifically, I started my PhD and I studied lionfish. And so that really got me more into venomous animals in particular, because I was really interested in understanding, the nature of their toxins, the genetics of their toxins, and how all of that works. And then after my PhD, I moved into a way cooler, in some ways, clade of animals, which is the jellies. So this here is a microscope photo of a box jelly tentacle. And what you can see there are all those little stinging cells wrapped up as little tiny cords inside those 90. And this is what happens after the tentacle hits a person, or in this case, actually, this was technically what happens after urine is put on a tentacle. And you can see all of those, so many of those 90 have now become sort of clear on the inside. They have discharged those tubules and delivered their venom to best they can. So if anyone asks whether you should pee on a jelly sting, the answer is no. Please don't. This is what will happen. And I have some videos here to show you what it looks like as best I can. They're not, Nat Geo quality videos or whatever, but here you can see some 90. This is from, I think, a Chrysora jelly. And now you can see that I have irritated it, and we see this unspooling of that thread, which will deliver the venom. If, your arm or whatever had been there, that would have been you. And then this is a Feisalia, so a Portuguese man-o'-war, some people call them, and a little bit of alcohol, and you get this. And what I love about this video is that you can actually see the droplets of the venom coming out of those tubules. And yeah. And then what I was doing for my postdoc was looking into understanding how we can treat jelly stings, in particular box jelly stings. And this is just a photo. This is one of the models that we use to investigate that. And what you're looking at on the left-hand side here, so this side here, those are actually red blood cells. So this is what we call blood agarose. It's a gelatiny sort of substance that has suspended within it a bunch of live red blood cells. So these are intact, healthy red blood cells. And then obviously we have our tentacle over here again with all those stinging cells that haven't discharged. But down here you can see that some have, and you see those little threads going into the agarose. And what has happened, the reason you have this sort of weird discoloration is that venom has lysed a bunch of the red blood cells. It is slowly diffused through the agarose, and it is lysed and destroyed, blown apart basically, the red blood cells in that area. So there's significantly fewer red blood cells, and that ends up making it look clear as that hemoglobin is released from them. And so this is a visualization of the damage being done by that tentacle, and the area of the cleared area is proportional to the amount of venom delivered. And so just another view of that. You can see this is what it looked like when I just put the tentacle on there. It was stinging as it was. And then this is, sometime later, you can see there are this sort of debris here is bits of the tentacle 90 that are stuck to it. And then this whole black area is where those red blood cells have been destroyed. And so we use this to understand sort of what treatments and what ways of interacting with tentacles after a sting can help reduce the damage being done by the venom. And it's very interesting because one of the things you might have noticed in the other slide is that there are a lot of 90 that still haven't stung. So when you get stung by a jellyfish, you have the opportunity to prevent a increase in the venom load or the amount of venom that goes into you, depending on what you do in that moment or in the moments afterwards. And so doing things like scraping the tentacle down, you're now encouraging all of those 90 to sting and things like that. So that's what I did for my postdoc. And I did not go unscathed in my investigations of venomous animals. This is an example. This photo I love so much because I'm trying so hard to smile. I really am. But it was right after I stung myself. You notice my bare hand here. That was a mistake. I didn't put on the gloves. You see in the little photo up here of the beautiful Feisalia that we caught. I now have my gloves on because I should have had them on before. But I managed to sting my thumb pretty badly while putting it into the bag. And so that is me grimace smiling, trying to look like I'm happy about this animal that just stung me. was the background for the book, was just my love of venomous animals and my interest in how their venoms work, how we can counteract how their venoms work, and what we can do to understand them better. I have never studied my favorite venomous animal, which is this one. For those of you who are unaware, that is the ankle spur of a platypus, and that I got to see up close and in person. That one for my book. I went and traveled to Australia and went and saw them up close. This adorable guy here, and you can see him, as cute as he is. He's right. This picture is him envenomating a towel because he was very upset about being disturbed. But they are one of the most unique venomous animals, and I see them as sort of a ambassador for venomous animals because it's unexpected that something so cute and cuddly looking, like you want to just snuggle them and play with them, would have such a disastrous venom, and their venom is brutal. One of the stories that I included in the book that I found in the scientific literature was of a former soldier who got stung because he picked up a platypus and he thought that it was struggling, and it was, at least it didn't appreciate being picked up, whether or not it was struggling. And it stung him in the thumb with one of these ankle spurs. And he said that the pain was worse than shrapnel wounds that he had from the war. He was in excruciating pain for days, and the only thing that alleviated it was a complete nerve block to his hand. So he was just unbelievable pain from such a tiny little cute critter. And that to me is why they're my favorite venomous animal. It's just unbelievably unexpected, and it just goes to show the incredible diversity of venoms and diversity of animals. And the fact that they deliver it with an ankle spur is even just weird, because most venomous animals use some sort of bite. As I said, one of my favorite parts of writing this book was in seeing the researchers firsthand, watching their research, checking out all the things they're doing, and getting to really watch what other people were learning about venoms. And here's me in the Amazon with a bullet ant, another one of my favorite venomous animals. And their name doesn't come from them kind of being shiny or like a bullet or the size of a bullet. It's because their sting supposedly hurts as bad as a bullet wound. And I considered finding that out for myself for the book, and then decided against it. Because nobody, that's not necessary. I decided that stinger there, whew, no thanks. Perhaps the one animal I was most excited to look at for the book was these guys. These are Komodo dragons on the island of Rinka. And part of the reason I became a writer and that I have loved writing was because I loved reading Douglas Adams's book when I was a kid, and in particular Last Chance to See, which was his exploration of endangered animals. And I always remembered and loved this chapter that he did on Komodo dragons. And so for me, being able to sort of follow in his footsteps and go to Rinka and look at these dragons, it was a dream come true. As for venoms in general, what makes them so amazing is that they are able to tinker and toy with our very basal systems in our bodies in all sorts of different ways. And this is a graphic from a researcher named Brian Fry and his papers, and it just shows how different animals tap into different aspects of the blood clotting cascade and in general hemostasis. And so you see, you've got all these different creatures having different effects on different parts of it to raise blood pressure or lower blood pressure or induce clotting or prevent clotting, and all of these things that affect. Similarly, there are dozens of different species and dozens of different types of toxins within those species that affect different aspects of our nerve signaling, and so altering and affecting the way that our neurons communicate. And these very, very basal systems in our body, these essential systems in our bodies, which in some ways is a little terrifying, but it's also very helpful for us. And a lot of these are being investigated now and have been, a number of them, turned into pharmaceuticals, because that's exactly what you want in a drug. You really want to have something that works in a small amount to do a very specific thing in affecting some important system in our bodies. The very first of these, of course, was captopril, which was a blockbuster drug, made billions of dollars. And it was basically an unaltered toxin from a viper, a snake. You also have you have lots of drugs coming out of leeches in their way of, altering blood clotting and coagulation. So there are lots of drugs that are used in things like surgery to make sure that you don't have clotting during a surgery. You also have animals like cone snails leading to painkillers because they have such potent and intriguing neurotoxins. One of my favorite drugs is Baeta, which comes from Gila monsters. And this one, it is somewhat of a mystery as to why this toxin exists in the Gila monster's repertoire. We don't yet understand what it does for the animal, but we know for us, it works great at helping deal with blood glucose levels. So another cone snail one that's in the works still is that the snails actually secrete an insulin, and it's really cool as to why they do this. The insulin is used to essentially put fish into an into a low blood sugar coma. They wrap their giant mouths around a fish and secrete this special insulin and knock them out. But it's different enough from our insulin. And what you can see here is the insulin of the cone snail is red and white, and the insulin in humans is blue, white, and green. And the lack of this sort of green part means that you have a really faster acting insulin, as it turns out. And so this is really, again, really useful therapeutically. And they're working on, I think they're still working on getting that pushed through clinical trials. And there are also a lot of weird effects from venom. This is a Brazilian wandering spider, Phonutria. And what you can learn about those guys is that a side effect of their bites tends to be priapism, so sustained erections. And so, of course, their venom is being looked at for erectile dysfunction drugs. And then just to finish it off, my absolute favorite venom-derived drug of sorts that is currently being worked on is from this. This is a Deathstalker scorpion. For those of you who aren't aware of scorpions, they are one of the only dangerous species of scorpions in the world that truly can cause harm to humans. And their venom contains a toxin that was originally called chlorotoxin because it was thought to bind to chloride channels. But they've realized it doesn't do that, and they have no idea what it does. But what it does do is bind to tumor cells, and almost exclusively to tumor cells. So Jim Olson over at the Fred Hutch Cancer Center, he's taken this peptide toxin and he's turned it into what he calls a tumor paint. And so he attached it to a fluorescent molecule, and you can inject this into animals. And now they're doing trials in children, particularly for brain cancers. And it will bind to tumor cells and then make them glow so that you can see tumor cells and tumor masses very easily under a microscope. So that to me is just like such an inventive and ingenious way of using a venom toxin in a non-toxic way, but in a very helpful way. Of course, no talk of venoms and venomous animals would be complete without talking about snakes. They are perhaps the most notorious venomous animals, and there might be really good reason for that. So one of the things I talk about in my book is really looking at venoms from an evolutionary perspective and trying to understand not just why the animals have their venoms in an ecological setting, but how that interacts with humans. And snakes are a particularly special group for this because there are some ideas, some hypotheses that some of their venomousness and some of our reaction to them and our fear of them is evolved, and that there is this long-lasting, it's not just because of, you know, cultural history, that there is this long-lasting evolutionary sort of arms race and battle between humans and snakes. And this is often called things like the snake detection theory. So one idea is that our eyes, and therefore our brains, in part evolved as a way of detecting snakes. And you can understand that many of these venomous snakes are kind of hard to spot. I mean, this picture is a little easier because he's so big, but if you were a little further away in this setting, this snake would be basically invisible, except to people, because we have these really good eyes for color vision, for seeing patterns, for spotting things like this. And I wanted to bring your attention to this mostly because there was actually a paper that came out at the end of last month, further providing evidence for this idea that of coevolution with snakes. And this was from Brian Fry and his colleagues in Australia. And what they did is they looked at essentially the innate resistance to snake neurotoxins, alpha neurotoxins. And what you find is that primate species that have evolved in the presence of these snakes, things like cobras, have a greater resistance to these toxins than primates that evolved in the absence of those snakes. And so just another sort of evidence for this arms race between humans and snakes in this long-lasting evolutionary battle. And I think that's really neat. And then lastly, just I guess I keep saying this, but my third favorite venomous animal, or my other favorite venomous animals, have to go to the parasitoid wasp, because what they do with venoms is just mind-blowing. And it is just frankly unfathomable that they would be able to do this kind of thing. This is an emerald cockroach wasp with her soon-to-be prey of sorts. So what they do is they sting and temporarily paralyze cockroaches so that they can sting into their brains. And they will actually probe the brain area with their stingers for minutes at a time if you remove certain regions of the brain. So these cockroach wasps, they have some sort of sensor in their stinger to tell them when they're in the right part of the brain. And they take that and they sting and they deliver it, and it essentially turns the cockroach into a willing participant in what's about to happen next. And so they're obviously, from the size difference here, they're not big enough to really drag a cockroach anywhere, but they can lead one. So they'll break off their antenna, they'll drink a little bit of their blood just to give them a little extra boost, and then they will drag and lead. So the cockroach is walking the whole time, like a dog on a leash. They will lead this cockroach to a burrow, and then they will place on that cockroach's leg a single egg, which will become the next generation of emerald cockroach wasps. And then they seal it in, and that egg hatches. It eats the cockroach from the inside out for its fuel source, and then finally the cockroach luckily gets to die, and the wasp will emerge. And it's just fascinating the way that they have these venoms that are so specialized to not just blatantly paralyze, but to dampen the will, essentially, of an animal. It's just unfathomably cool. And so that's just a sampling of my favorite things from within the book that I wanted to show you guys. And with that, I'm done, and we can get back to talking.
All right. Thank you so much, Christie. That was fascinating. I especially like how you kind of updated a lot of the threads, that you explored in the book since its publication. That's really cool with regard to research. So before we dive into, is that my microphone? Sounds like the ocean. Oh, there we go. Before we dive into the Q&A, I just want to remind the audience, don't be shy. You can ask any questions you want within reason. Using that Q&A tab, and share your thoughts with us, and we'll pose them to Christie as we can. We've already got a couple good ones. I do want to, as I commonly do, invoke the moderator's privilege and ask the very first one. And Christie, I just want to preface this with a bit of a disclaimer.
Okay.
So what I'm going to say, any resemblance to actual events, past, present, or future, is purely coincidental. But I'm curious, if you had to kill someone, how would you do it? Which snake would you use?
Wow. If I had to kill someone, what? So if I had to kill someone, see, that's so tricky, because you got to question whether or not you want speed or your own handling of the animal, right? You're going to have to wrangle something long enough to get it to do its thing. I don't know. I still think killing people with a blue-ringed octopus is my favorite idea. So blue-ringed octopus they're teeny. They're like hand size, so they'd be travelable. Like you have to have water, but you could still kind of travel with them fairly straightforwardly, I think. And then, but their venom contains tetrodotoxin, so it's one of the most potent toxins we know of in the world. And so if you could you would just get them to hold this cute little teeny octopus, and then they do something and it bites them, and then they would die. And so maybe they wouldn't even realize that they were in trouble. There are so many videos, if you go search on social media, there's so many videos of people being like, oh, look at this cute little octopus, and I'm like, don't die. So maybe you could, not even look like you're trying to kill someone.
Yeah, and I think you talked about in the book with regard to the blue-ringed octopus, was it them that there's some thought that they may have co-opted their venom from like a bacterial symbiont or something like that? Is that right?
Yeah. As far as we understand it, we don't know of any animals that can produce tetrodotoxin themselves. So the idea is that all of the animals we know of that have it, so things like puffers, things like certain newts or salamanders, all of them are getting it from somewhere. And the idea is that that's like certain species of Vibrio or other bacteria.
Cool. So I want to get to some questions from the audience. And I think, so this first one is from Norman in Toronto, Canada. And I think he's referring back to your picture of the Portuguese man-o-war and your sting on the thumb. So Norman asks, should you have sucked on your thumb to reduce the venom?
No. So when those tentacles graced across my thumb, what happened is that these 90 shot out those tiny little threads that you saw in that, especially in that second video. That's the same species. And those tiny little threads pierced into me and were delivering venom. And so there was nothing I could do at that point to those threads. Like, they're too small for sucking to work to pull it back out. Like, I could deliver something into my skin that could counter out the venom toxins, but short of that, like, there's nothing I can do to stop that venom from being delivered. The key thing is that not all of the 90 on my skin or on those tentacles went off. And so there were likely a number of undischarged stinging cells. And those have the potential to discharge if you do something to them. So if I had put my mouth saliva on there, that would have almost definitely aggravated those stinging cells, and they would have stung my tongue or my finger more. And that would have been very, very uncomfortable. The best thing to do, which is what I did, is I rinsed it off with vinegar, and then I used heat. So I had one of those little reusable heat packs, like you get for like skiing or whatever. And that reaches a temperature of about 45 degrees Celsius, which is, as hot as the human body can stand nearly without scalding. And so I heated it, and I applied a experimental treatment being developed by Angel Yanagihara, who is my postdoc advisor, in Hawaii, called Sting No More. And so that, once I applied that cream, it started to feel a lot better, because that actually contains things that mess up the toxins in the venom.
Yeah. Cool. So now more of a comment, but it kind of leads into a question that I have. Sorry to hog the questions. But Elaine from Silicon Valley says, fantastic discussion about the biomedical applications. I'm so thrilled you talked about the cancer imaging agent from Blaze Biosciences based on chlorotoxin for use in pediatric glioblastomas. That's my niche specialty, and I can't believe this is showing up in this talk. Awesome, exclamation point. So I wanted to ask, you kind of updated us on a lot of the biomedical pharmaceutical stuff. You also talked in the book about animals who specialize in eating venomous animals. I'm wondering if there's much research going on in that kind of realm, studying those animals and their resistance to those toxins and how that could inform like antivenoms and things like that.
Absolutely, absolutely. And there definitely is a lot of research. The tricky thing about resistance to venoms is it depends on how the animal is resistant. So there's really two main ways that an animal can become resistant to a toxin. They can change whatever that toxin is targeting and just make it so it doesn't work anymore. The idea here being like if you have a neurotoxin that, you know, blocks some sodium channel or something, you can have slight changes to the amino acids or whatever of those sodium channels and make it so that doesn't bind anymore. All of a sudden the toxin doesn't work. That's hard from an evolutionary standpoint because, again, these things target really basal systems in your body, and you don't want to mutate your sodium channels so that your actual nerve signal, you know, don't work on them anymore, things like that. But there are animals, mongooses are a perfect example, that seem to have done exactly that. There are others that have done really cool things where they've added like a bunch of sugars to the outside, so it's just trickier to get these generally larger venom toxins in there, as opposed to the like small molecules that the body uses. And there's things like that. So you have a bunch of cool sort of modifications to the target. That's really hard to translate clinically because we can't just go around gene editing everyone's nerves or whatever to make it so that they can't, you know, be, you know, bitten by a snake or whatever. The other way to be resistant to a venom is to have some sort of circulating molecule that inactivates the venom or, you know, binds the venom or something like that. And that we also see in resistant animals. So things like opossums, and they have these serum proteins that bind up toxins from snakes. And so they're highly resistant to a lot of snake toxins. Now that is less complete a resistance because theoretically speaking, one could get overwhelmed with just too many toxins if you don't have enough of those defensive molecules. The toxins still make their way to where they need to go and do their thing. But that is essentially the idea of antivenoms anyway. So antivenoms are just consisting of antibodies that are, you know, binding to toxins and binding them up and hopefully neutralizing them. And so there is a lot of research looking into sort of these transferable, as they call them, resistance mechanisms and whether or not we can create, you know, a pill for snake bite or something like that contains these transferable resistance mechanisms.
Yeah, staying on that topic, Elaine asked kind of a follow-up question. She says antivenom production for certain substances has dwindled or been completely stopped over the decades. Thoughts or comments?
Yeah, so there are some particular cases, especially I think with certain species of African snakes where the antivenom production has dwindled or stopped. And a lot of that is unfortunately political. It's not so much that these antivenoms weren't any good or that they didn't work. It's that it's an expensive drug to produce. You're talking, at least as of now, all of our antivenoms are produced by animals. So you have to raise animals, you inject them with certain amounts of these toxins, and they produce a natural antibody response to it. And then you collect those antibodies by collecting their bloods and serum, and then you purify them, and that's it. That's your antivenom. And so the whole production process is extremely drawn out. It's expensive. It's hard. And so for certain pharmaceutical companies, it's just not worth it, especially if you're talking about poor regions where getting these antivenoms is difficult, affording these antivenoms is difficult, storing these antivenoms is difficult. They tend to have to be kept cold, like refrigerated, and not all places that have a lot of snake bites have a lot of refrigeration capacity. So there's a lot of sort of socioeconomic and political reasons why antivenom production has decreased. Now, that said, there are a lot of people trying to fix this problem, in part by trying to do new ideas for producing antivenoms. So cell culture produced antivenoms, for example. If you can get those antibody sequences into like yeast or something that can produce a whole bunch of antibodies in a test tube, then you don't have to have the whole animal production thing. You might be able to cut production costs. There's also, altering those antibodies to make them more easy to produce or smaller, for example. So there's a lot of work being done trying to create sort of the next generation, they call it, of antibodies that are easier and cheaper to produce and, shelf stable, so that you can just send them places they can sit on a shelf for days and days and days on end and then be used as needed. Things like that.
Cool. So I want to get to some sort of behind the scenes questions that are coming in from listeners. So Melly from the Great Lakes, somewhere around the Great Lakes, says, fascinating work, Christie. What type and how did you secure funding for all this work, including travel around the world? Was there a book advance?
So yes, there was a book advance. My book was published by FSG, and specifically Scientific American's publishing arm. So there was a book advance that helped. I also secured some funding for sort of spin-off stories, essentially. So I wrote a story about venomous animals that involved travel, and so that I was able to go to these places and then also talk to people for my book. The one that comes to mind, for example, is I wrote a story for Mosaic, which I think is no longer around. It was a Wellcome Trust publication or funded publication. And so I wrote a piece on venom drugs, basically drugs from venoms for them. And that involved traveling to Australia.
Awesome. So here's back to the science. Robert asks a question. Is there an evolutionary basis for classifying venomous creatures according to whether their attack is for defense rather than for hunting?
So there are some interesting differences between predatory and defensive venoms in certain ways. Absolutely, there are certain similarities between defensive venoms and between offensive venoms. For example, defensive venoms tend not to be as lethal because, well, they're not really trying to kill anything. I mean, that's not the purpose of the defense. Instead, they tend to be really, really pain producing and really quickly and actively pain producing. And so you have something like a stingray's venom hurts a whole lot, but the venom itself, not really lethal. I mean, no one, as far as we know, has died from the venom. They've died from the barbs, if being put into sensitive body parts, because you're talking about like a 3-inch serrated blade. But if those blades never went near, heart and livers and things like that, then those animals would never be able to kill a person.
All right. A question from Russ in Chicago. Russ Ziegler, I think is how you pronounce it. He says, with the scorpion, where you do not know how it kills, where do you start to learn how the venom itself works?
Yeah. So something like that, we have some ideas for how the venom as a whole works. You can do studies in mice or other animals, for example, and you can separate out different parts of the venom using something like an HPLC, and you can start to tease apart what different things do. Like, do they slow the heart? Do they kill cells? Do they do these different activities? And so you have these different, they call them assays. So you have these different ways of looking at what the venom does. But in this particular case, what it was is that we didn't know, we still don't know what chlorotoxin, that particular peptide does, other than we know that it tends to bind to tumor cells. And so like, we're still trying to figure out, like, just putting it on cells and seeing what happens, knocking out. One of the things that I assume might happen in the near future is something like a CRISPR screen, where they'll take a bunch of cells that are tumor cells or whatever that are susceptible to it, and they'll start knocking out genes one by one with CRISPR, and by them, do 1 million different genes, and then see which one becomes resistant to the toxin. And so which which gene is this toxin interacting with in some way, or which genes are they being interacted with?
Yeah. So Pamela asks a wardrobe question. Pamela is curious about your necklace pendant. Any background story?
Yeah, I love this. So I bought this from an artist in Maui, and I actually gave a matching pendant to my postdoc advisor, Angeliana Gihara, and so it was sort of it was just a I don't know, a cute lab thing that I saw it and I was just like, I need to own some sort of jewelry. I actually have jellyfish earrings on too, and a jellyfish dress, because you gotta dress the part.
I like it. So Jim Elder asked a question that you actually addressed in the book. Jim asks, can individuals, for example, zoo snake handlers that have been bitten several times eventually develop resistance? And this is where you talk about that whole crazy world of SIers.
Yeah. So there is some evidence anyway that, yeah, if you get bit multiple times or you experience the venom in some way multiple times, in the case of these self-immunizers, they're injecting it intentionally into themselves in small doses, that you can build up antibodies, and those antibodies may protect you in the case of a future bite. Just like what we do for antivenoms, really. It's just the idea that you expose to these toxins, your body does produce antibodies, and then you have you have potentially resistance to it. The tricky part, or at least the part that's really difficult when it comes to something like snake toxins, is that, for one, unlike an infection where you start with just like one or two little, small amount of this infectious particle, and it has to replicate a lot before it becomes really a problem. So your body has time to kind of find it and locate it and get rid of it with antibodies. With venoms, you're talking about a huge dose, or what is, for your body, a huge dose potentially of these toxins at once. And so for your body to actually be resistant to that and not experience the ill effects of it, you have to have a lot of these antibodies already circulating. You have to have them on the go, ready to go. And so in a lot of cases, that's difficult. That means constant exposure, because your body, what it'll do to something like these toxins is it'll circulate the antibodies right afterwards, but then over time the number that are circulating will go down, and you'll just have those memory cells that are ready to produce when they see this toxin again. And usually with something like a snake, that would be too late. Like the amount of time it would take you to ramp up your antibody production would be too late. So you have to basically be constantly exposed to the venom, which is what self-immunizers do, to their credit, is they regularly inject the venom so that their antibody levels stay high. Doesn't seem terribly practical to me. It also seems extremely dangerous, because you mess up on that dose and all of a sudden you could overwhelm those defenses and be in serious trouble. But there is some investigation into using those antibodies that people have developed to develop more human antibodies. And so one of the issues with antivenom, longstanding issues, is that you're talking about proteins from a horse or proteins from a sheep or something like that. When those proteins are injected into a person, there is a not insignificant chance that they will have an allergic or severe immune reaction to the proteins, so to the thing that is supposed to be defending them against these venom toxins. And so it's one of the reasons why antivenom is basically only administered in hospital settings, because you need to have someone there to be like, okay, wait, they're going into anaphylaxis, we need to deal with that too. It's usually still worth it. Like, you still want to have the antivenom because it's going to do its job, even if you have to tamp down your immune response to it to let it do its job. But it just complicates things. So if you had human proteins, if you had human antibodies, your body would be less likely to see those as foreign and less likely to react to them. And so that might be one way to help make antivenoms more affordable and accessible, is because you could theoretically administer them in less hospital-y settings.
Yeah. So Sue Kin asks a question, and this has to do with one of the more fascinating aspects of your book, I think, and that was thinking about like how these types of mechanisms evolved. So Sue asked, do venoms in different species of an animal in the same genus have the same mode of action? And then as a follow-up, Elaine asked, Elaine from before, do venoms show convergent evolution, that is, different structures and molecules that do the same thing?
So the first answer is yes and no. So often you see this sort of phylogenetic signal where you see that closely related species have similar venoms, but it's not always the case. And in many, often there's actually individuals of the same species can have very different venoms. And so something that's a particular issue, I'm trying to remember the species. I want to say it's Mojave rattlesnakes. Some rattlesnake species in the Southwest where the venoms are very, very different, even on the scale of miles apart. The inter-individual venom variation is just huge. And scientists think it might be related to diet, it might be a little bit related to habitat, but some of them just have tons of neurotoxins, while others have tons of sort of hemotoxins, blood targeting toxins. And that poses a problem when you're looking at something like an antivenom, because if you have different toxins in there, you don't know which one the antivenom is going to necessarily counteract. But yeah, so there is this ability to have variation, and especially because you have these venoms aren't one toxin. Very rarely are you talking about one toxin being a venom. Something like bees is probably the closest to that, because you have things like melodin, which is 50+% of the venom. But even then, you still have this like handful, this like grouping of other toxins that are thrown in there that make up that other, 50-40% of the venom. So these are cocktails. And so an individual or different genera, as related, can sort of ramp up or down the production of this suite of toxins that they have at their disposal. And so you can have different mechanisms of action even within a species, but certainly closely related species can have different mechanisms of action as well. Another place we see that is in cobras. And so cobras are notorious for neurotoxins. Their bites are usually considered basically painless. And you don't even know. People talk about like not even knowing they're bitten at night while sleeping, and then just dying because they get paralyzed. But spitting cobras have a lot of more pain production and other things in their venoms often, and that's probably an evolutionary thing, so that when they spit their venom and it gets into your eyes or your mouth, it does something. It has this effect on you, so that you're feeling it. Whereas if you spat a bunch of painless neurotoxins, like, the predator isn't going to know that there's a reason not to mess with that snake. So you can see things like that even in closely related species where there's been this pressure to alter the venom composition. As for the convergent evolution, the answer is yes. I mean, venoms by and large converge not only in just action, so things like paralysis, or they attack the same targets, and even you see repeated recruitment from the same gene families. And so you might see one of the more common ones, phospholipase A2, and that's an enzyme that cuts fats, and the fats that it cuts off tend to do nasty things, like they signal inflammation, they signal these other things. And so PLA2 enzymes are seen in basically, I'm trying to think of a venomous group where they're not seen. They're seen in everything, like they're in insects, they're in spiders, they're in snakes, they're in pretty much every major. I think they're in scorpions. I mean, you've got these PLA2 enzymes being recruited over and over again for venomous purposes.
Yeah, excellent. I want to return to the SIers for a second, because one of the things that I really liked about the book is that you not only introduced us to kind of a fascinating menagerie of animals, but I think the human cast of characters was almost equally interesting to me anyway. And it seemed, and forgive me, I don't remember all the names, but I mean, you had like the mild-mannered rattlesnake wrangler, and then like the kind of crazy Australian guy, and like, everybody in between. But I think a common thread may have been, and I'm curious to hear your perspective on this, may have been like people who were a little bit kind of enamored with danger, like adrenaline junkie type people. Did you see that thread, or do you see that in the kind of collection of people that study venoms? And even more importantly, do you see that in yourself?
I definitely think that there, especially people who study some of the deadlier animals like snakes, there tends to be it's at least a frequent trait. Let's say that. It's not universal, but there is definitely a little bit of it helps if you're the kind of person that sees a snake and doesn't want to back down if you're going to be trying to collect venom from a snake. And then so you've got a lot of people that have an affinity for danger and dangerous creatures. But you also have people, I mean, not to bring her up again, but my postdoc advisor, Angel, I mean, she is not a thrill seeker in my book. Like, she is a very sort of kind, gentle sort of person, but she works on box jellies because she almost died from a box jelly sting. And so she had this curiosity spawned by a life-threatening event. And that's just dictated her entire career, basically, is this fascination of, at the time, like, this was the late 90s, we had no idea what was in those venoms. They were too hard to collect. They were too hard to really to study. We didn't have as good a genetic or protein tools that we do now. And so she wanted to know why she was feeling the way she felt. And she didn't get any answers from the scientific literature. So she decided to find out for herself. So you have a lot of people like that in there too. And you have, a lot of doctors that, who went into being doctors because they love people and they wanted to help people. And then, they're seeing firsthand, the morbidity or mortality from something like snake bites in rural Africa. And they want to do something to figure out how to stop that. And so you do end up having a pretty good diversity of people. As for myself, I plead the fifth.
Never been skydiving?
I have been skydiving.
All right. No more needs to be said. So we're getting some fascinating questions from the audience. Thank you so much, everyone, for participating. Just to remind you, on the right-hand side of your screen is that Q&A tab where you can submit your questions. So Joseph Farley asks, there's been some speculation that snake toxin sequences, for example, BGTXs, can end up in viruses, for example, SARS-CoV-2, through recombination events. Comment?
I haven't seen any really credible evidence of viruses infecting humans and snakes sort of equally in that sort of way. Something like SARS-CoV-2 really is better at infecting mammals by and large. So it's less likely or very unlikely for it to infect a whole bunch of reptiles and then have that kind of recombination opportunity. There were some original thoughts that maybe it was a snake toxin. I remember from way back in early 2020, but they were based on just the types of codons and then the types of amino acids basically being used in certain proteins and they ended up being pretty shaky. So I've never seen a snake venom toxin then being employed by a virus.
Okay, fair enough. I want to get to this one because it sounds like it could be urgent. I hope it's not, but I think Mira was her name from before in the Great Lakes, says, could you please walk us through the immediate practical steps to take in case of a venomous bite in an outdoor setting till and if reaching a medical facility? So I hope that's not happening presently, but if you could walk us through.
So from what I know, and I again preface this, I am not that kind of doctor. But one thing you want to do is sort of try to immobilize it. You don't want it pumping this venom through your body any more than it has to. So if you're bit on the foot or something, you don't want to like wiggle your toes around. You want to like try to immobilize it, try to keep it lower than your heart. Again, trying to keep those venom toxins sort of where they are. Doctors don't usually recommend a tourniquet unless you're a doctor who knows what you're doing, because you're more likely to just cause your problems, other problems to that area from cutting off blood flow, and just really get to medical attention as quickly as possible. You're not going to be able to suck the venom out. You're not going to be able to pull it out. They have those devices. The people I know who study snake venoms hate that those things are sold. They're like, it's very different than something like a bee sting, where you do want to pull the stinger out because the stinger is sitting in there, and it isn't like, you can actually see it if you look at videos. It's sitting there pumping and putting more and more venom in. So yeah, with a bee sting, you want to remove the stinger, and you do kind of want to like that suction action for that. but not for a snake bite. It's not going to happen. It's already injected deep into your blood and into your tissue. You're not getting it out. And so you just want to get to a doctor literally as fast as possible.
So Hollywood has led us astray yet again. You don't, if your companion gets bit by a rattlesnake, you don't suck the venom out.
You don't want to. And even if it, like, technically, even if you could, let's just say hypothetically you could, all it would take is like the tiniest cut in your mouth and you'd now have a snake bite in your mouth.
Yeah.
Like, this would be bad.
Yeah. Hear that, everyone? So Jim Elder asks a very interesting question. And this is something that I was interested in kind of thinking about as I read your book. He asks, is the venomous evolution primarily parallel evolution, or does research suggest there was an original primordial venomous creature?
Oh, man. So I think what we see in terms of like the creatures that we consider to be potently venomous now, you're seeing convergent evolution. That said, there is a very healthy and active debate amongst herpetologists as to whether or not in reptiles there was a sort of early venomous ancestor that then split. That's the Toxicophera hypothesis, I think is what it's called. But the idea is you have certain venomous lizards like the Komodo dragon. You have certain snakes like, vipers and elapids like cobras. But then you also have, iguanas, all these and boa constrictors. So you have these other snakes and these other lizards that aren't venomous. And the question is, did the two venomous groups there evolve separately, or was there a venomous ancestor that then lost its venom for a number of the different groups? I don't know that the researchers have settled on a consensus there. I think there is some decent evidence that even species like iguanas have some of these capabilities for venom, or these like certain gene duplications or whatever that would indicate that they once may have used something for a venom, but it's still, jury's still out on it.
Yeah, I mean, certainly since being venomous is so widespread across animals, you wouldn't think that there's like a single ancestor that was like some kind of like unicellular organism or something that kind of gave birth to all those venomous species. But yeah, I think that it's interesting to think about within those different kind of branches, reptiles, mammals, insects, there may have been kind of like a nodal ancestor that was venomous.
Yeah, and what's interesting too is that it's really a question of when do you consider something venomous? Because a lot of these proteins, like you and I have PLA2 enzymes. Like we have them. They may even be in our saliva. Like we have them in places where one could potentially develop a venom. And so where do you draw the line between something that has a slightly potent enzyme in its mouth, that it uses to bite, and something that is a truly venomous creature? Somewhat maybe a bit of semantics, because again, these are all things that they produce by themselves. So they are things that are frequently common across animal groups. So that's just good for them.
I have another question that I was curious about as I was reading the book and kind of knowing you and having worked with you for the past several months. And you talked about it in your presentation here, like you have lots of favorites in this world. I'm curious if you could kind of like, take off tomorrow, what would be the next kind of venomous quarry that you would pursue? Or would you maybe revisit one? I mean, I think the scene that you painted of like Rinka and seeing the Komodo dragons, you obviously took great delight in that. But if you could go like tomorrow, literally, and have, a paid flight somewhere to go chase down a venomous critter, which one would it be?
It's so hard to choose. I think I'd have to go for something I haven't seen, just because, I mean, as amazing as the ones I've seen are, I think I'd want to like keep diversifying. One of the ones that I really like that I've seen videos of, but I've never seen in person, are some of the spider-tailed vipers. And I just think that would be a really cool thing. I don't know if you guys have never seen this, look up spider-tailed viper, but they basically have their tails, instead of something like a rattle or whatever, they have this appendage. It's got like a couple of segments that are bulbous, and then it's got a bunch of little frilly bits. And when they move them, I mean, dead ringer. for a spider. I mean, it looks like a spider, and they use it to lure birds, and then the birds come down, try to peck at the spider, and then the viper kills the bird. But that would be unbelievably cool to see in person. So I'm gonna go with that.
All right, good answer. So I'll get to another audience question. Jeffrey Sperber asks, are poisonous creatures, now there may be some semantic stuff to sift through here, but he says, are poisonous creatures immune to their own venoms?
So this is a fantastic question, and it's one that I love answering, because the answer is sometimes. So we do know that, for example, cobras tend to have their neurons, the channels on their neurons, for example, tend to be resistant to cobra venom toxins. And so you see actually convergent resistance in something like the mongoose and something like a cobra. They have very similar changes to their sodium channels to make them immune to these venom toxins. So yes, in some cases. In others, no. And we actually have reported literature cases of, one was a brown tree snake, for example, in Guam, I think it was, that bit itself when it got captured, and it died like shortly thereafter. So there are definitely ones where the only thing keeping that animal from dying from its own venom is the fact that it is, being cordoned off in this gland or whatever, and not being exposed to the general bloodstream all the time. So yeah.
Yeah, and I would imagine that especially becomes important in cases like the platypus, where those venomous spurs are used for like fighting in males, right? So
yeah,
obviously there would probably be some effect of that venom.
So there might be some resistance compared to a person when it comes to the platypus venom, but the platypus and the venom is only in the males, I should clarify. But male platypus have been known to kill each other. If you go talk to a zoo or whatever in Australia that displays them, they have to keep them separate because they will kill each other.
Yeah. So Elaine is back with another question. This is an important one. Elaine asked, to purchase the book, I got my copy from the Webster Groves, Missouri Public Library, but to purchase the book, should we go through Amazon, or is there a better way?
I mean, that probably has your own personal opinions of things like Amazon. I'm always a big fan of small bookstores and local bookstores. So if you can find it or order it through your local bookstore, that's what I would recommend.
Okay. I want to ask kind of an emotional question, and it has to do with kind of your process of writing the book and how it affected you. Obviously, you're someone who is fascinated with nature, right? Did learning about how awful some of these species can be in relation to humans, I guess, like the caterpillars that make you bleed from your eyes, for example, did that kind of alter your did it make you more fascinated? Did it kind of inject some kind of like, I don't know, just some macabre kind of like flavor into your fascination with nature? To me, it kind of makes it seem like, I personally think that humans are, if you look at animal species, humans are pretty horrible in terms of the things that we're capable of and things like that. But a caterpillar that will make you bleed from your eyes and everywhere else is kind of like worse in a way.
Yeah, I think, well, first off, if I wanted to harm the person I'm supposed to murder and make their life, like their end of their life, as painful and horrible as possible, I would definitely choose that caterpillar, the Nomia. But anyway, other than that, I think for me, honestly, it's just more fascinating. It's hard for me to hold it against them. It's sort of like when people talk about tigers and their big claws and their big teeth, and it's hard for me to hold it against them. It's how they evolved. It's this ingenious, frankly, strategy, and especially when it comes to venomous animals, they're ingenious. Like they are doing and have done for millennia what the best biochemists in the world can only dream of doing, and finding and designing essentially through evolution these molecules that are just incredibly good at doing certain things. So for me, I'm endlessly fascinated by all of it, and I can't imagine being not fascinated or holding it against the animals that they can do these terrifyingly terrible things.
Yeah. So you had personal run-ins, right? That some of which you talked about. You talked about the urchin spines in Hawaii. You talked about the Portuguese man-o-war. a member of the editorial team here at The Scientist actually asked, how do you ever go into the ocean, knowing what you know about all the venomous animals that live there? There was the fish in the cave. So.
It's to me, it's sort of like knowing that sharks exist, but still going in the ocean. These animals, these creatures, their goal isn't to sting you. You're not food, unlike a shark. You're not potential food for any of them. You're not, the only thing you would be is someone that is going to cause harm to them. And so they would be just defending themselves if you did something. I do shuffle my feet when I'm in the water off of South Florida, because I don't want a stingray or a stingray barb in me. I learned the stingray shuffle a long time ago for that reason. And I'm cognizant. Like if I go diving on a reef, I'm careful not to touch the urchins now. I was careful before, just that one particular instance. So yeah, they're not out to get you. So it's for me, it's pretty easy to go share the water with them.
Yeah. There was another question of a kind of logistical nature from Louise. It may have been answered in the Q&A, but she asked, will the book be coming out in audiobook format? I think it's already out, right?
I think it is out in audiobook format. I believe so.
Yeah. I have to ask you the typical kind of book club author question, like, when's the next book coming out? And what's it going to be about?
I do have an idea for another book. I guess as my boss, I'm not sure when the next book will be coming out. I don't know what I want to say here. I do have an idea for another book. I've started working on a proposal, but no firm plans yet.
Does it involve putting yourself in great bodily danger?
Kind of.
We'll talk. Well, I have to, Elaine has been a very consistent and vigorous contributor, so I have to just put in a comment from her. She says, as a scientist myself, I am so impressed and amazed by both the depth and breadth of knowledge you have on pretty much every topic, species, and question that we've thrown at you. So thank you, Elaine.
Thank you.
Yeah, and I think that's basically all we have time for. It's been fascinating, Christie. I really thank you. And on behalf of the audience, I thank you for sharing your experiences and your thoughts and your book with us. As a reminder, this webinar will be archived on the Scientist website, and you will receive an email notifying you when the on-demand webinar is available. Please also feel free to comment on that aforementioned social club on our Facebook page, the Scientist Facebook page, and give us some feedback on how you thought this event went. So if you have any further questions, you can reach out to Christie directly. Her email and her Twitter handle are listed there on your screen. And I would like to personally thank everyone who took the time to join us today, and particularly those of you like Elaine who shared your questions and comments. Christie, it's been a pleasure getting to know your work from before you were a member of the Scientist, and it's been a pleasure working with you ever since. I hope it continues for a long time. So thanks again to everyone for tuning in. Thank you, Christie, for participating. Everyone, please have a peaceful holiday season, and we'll see you next time. Take care.